Hello guys, today we are going to see about the life cycle of Gnetum. Gnetum reproduces sexually. It is a dioecious plant, so it contains both male and female inflorescence. The male inflorescence contains the male flower and female inflorescence contains the female flower. Both inflorescence are compound. The male flower gives rise to the microsporangia. The female flower gives rise to the megasporangia. Both microsporangia and megasporangia use sporangiate. The microspores are formed from the microsporangia and the megaspores are formed from the megasporangia. The male gametophyte is formed from microspore nucleus, where the female gametophyte is formed from megaspore mother cell by meiotic division. After the formation of male and female gametophytes, it undergoes pollination, where the pollen germination takes place. After pollen germination, the fertilization takes place. The male and female gametophytes fuse together to form zygote. The zygote undergoes the embryonic development to develop the seed. After the development of the seed undergo seed germination to produce the new saprophytic plant body. Development of microsporangia. Microsporangia is a eusporangiate, so it is developed from a single initial cell. Which is microsporangial initial cell. It divides periclinally to form other primary wall cells and multilayered sporangial wall. Those wall cells undergo repeated periclinal and anticlinal division to form multilayered sporangial wall. Later, these cell walls organize themselves, forming the outer and middle layer of the sporangial wall, which are called as epidermis and parietal layer. The inner tapetal layer is formed from sporangineous layer. Archisporial cells divide repeatedly to form sporangineous cells, which form the central sporangineous tissue, which behave as microspore mother cells, and undergo meiotic division. It forms the tetrads of haploid microspores or pollen grains. Spore mother cells represent the lost cells of the sporophytic generation. Middle layer and tapetal layer of the microsporangium disintegrate for providing nutrients for developing microspores. Haploid microspores or pollens are produced in the microsporangia of the anthers. At maturity, the anthers degases by longitudinal slits to release pollens. Development of megasporangium. It is also a eusporangiate. Annular meristem develops below each collar. 8 to 10 cells differentiate as megasporangial initials. These are born on a cushion. This megasporangial initial undergoes repeated division to form homogeneous mass of parenchymatous cells or nucleus. Cushion undergoes repeated cell division, gives out 3 envelopes all around the nucleus. The ovule is formed. Gametophytes. Microspores and megaspore represent the first stage of gametophytic generation, and they develop to male and female gametophytes respectively. The female gametophyte developed from the megaspore mother cell. Megaspore mother cell undergo reduction division to form 4 cells, which may become an embryo sac. When several megaspores begin to function, several gametophytes may be produced within a single ovule. No archegonium is present. Male gametophytes are produced from microspore nucleus, which divides into 2 daughter nuclei and later divides again into a tube nucleus and a generative nucleus. Thus, 3 nuclei are formed: prothallic nucleus, generative nucleus, tube nucleus. Microspores are shed at this 3 nucleate stage. Further development takes place in the pollen chamber after pollination. Stalk cell remains, only body cell and tube nucleus enter the pollen tube. In this, we can see the different stages of germination of seeds.